Today we're going to have a look at hormones. In this session, we will look at methods for measuring hormone levels. We will start by looking at what hormones are and will aim to understand the different types of hormones and how they work. We will then consider why as psychologists we would want to measure hormones. We will consider hormones that are involved in homeostasis, including thirst, hunger and the sleep-wake cycle, and the hormone cortisol, which is involved in the stress response that we might see in anxiety and phobias and other stress-related situations. So what are hormones? Well, a hormone is a chemical released by a cell, gland or organ, which travels through the body, mostly in the blood, to influence the cells in another part of the body. Hormones are similar to neurotransmitters or neuromodulators, except that they act over much longer distances. Hormones can be either steroids or non-steroids. The endocrine glands synthesizing steroid hormones develop from different embryonic tissue than the glands secreting non-steroid hormones. The glands derived from the mesoderm, consisting of the adrenal cortex and gonads, produce steroid hormones, while those derived from the ectoderm secrete non-steroid hormones. The steroid hormones are synthesized from cholesterol in the adrenal cortex and gonads. The adrenal steroids are glucocorticoids and mineralocorticoids, and the gonadal steroids are progesterones, androgens, and estrogens. The non-steroid hormones can take many forms and include modified amino acids, which are hormones synthesized from single amino acids, such as thyroid hormones and indelamines, for example melatonin and serotonin. Non-steroid hormones also include peptides, which are hormones formed from chains of amino acids, such as oxytocin, which is a small peptide, glucagon, or glucogen, which is a large peptide, and insulin, which is a protein or polypeptide. Non-steroid hormones can also include glycoproteins, which are hormones synthesized from proteins and carbohydrates, such as follicle-stimulating hormone. And non-steroid hormones can also include prostaglandins, which are synthesized from essential fatty acids. A large number of illnesses, disorders, conditions, and clinical states exist which are associated with over or underproduction of an imbalance in the levels of circulating hormones, particularly steroid hormones. Hormones that affect behaviour through neurons do so by altering the activity of particular groups of neurons in the brain. For example, the sex hormones have important effects on behaviour. There is a reciprocal relationship between the level of hormone and psychological factors. For example, testosterone levels are related to degree of competitive behaviour and aggression in males. Defeat of male primates leads to a fall in production of testosterone. Losing a tennis match or wrestling competition has also been shown to cause a fall in blood levels of testosterone. And this shows that psychological factors can have a direct effect on hormone levels. So as psychologists, hormones are of great importance to us. So how do they work? How do they influence behaviour? Let us take the endocrine glands as an example. Hormones are synthesized and stored in endocrine cells, and on cell stimulation they are released into the circulatory system. Hormones stimulate receptor molecules on the cells in target tissue, including neurons. Almost every cell of the body contains hormone receptors of one kind or another. Hormones can only act at cells which have receptors for them, and receptors are specific for particular hormones. For example, estrogen will have no biological effect on a cell unless that cell has an estrogen receptor. Hormone release occurs when the endocrine cell is activated by electrical, neurochemical, or hormonal stimulation. Once the hormone is secreted into the blood, it is likely to be rapidly deactivated. To prevent this deactivation, many hormones are bound to protective carrier proteins in the bloodstream. The carrier proteins facilitate transportation of the hormones in the blood and prolong the active life of the hormones by protecting them from deactivation. While the hormone is bound to the carrier protein, it is inactive. Thus, high levels of protein-bound hormones can be held in the blood to act as readily available storage reserves. Before a hormone can bind to its target cell receptors, it must be uncoupled from its carrier protein. The hormone carrier protein binding is reversible, and this bond is weaker than the hormone receptor bond. Hormones can, therefore, exist in two forms, free or bound. Only the free hormones can attach to a receptor at a target cell. Carrier proteins help to regulate the level of free hormone available to the target cells and thus protect the body from excessive hormonal stimulation. Hormone levels in the bloodstream fluctuate quite dramatically over short periods of time, yet they are maintained within certain average levels in the long term. The regulation of hormone levels in the bloodstream occurs through the autonomic and central nervous system, non-hormone chemicals in the bloodstream, and gastrointestinal tract and hormone feedback. So there are many mechanisms which are controlling hormone levels within the bloodstream. Hormone synthesis, storage, release, transport, and deactivation occur through a variety of different mechanisms depending on the chemical structure of the hormone. The hypothalamus and the pituitary gland are both highly involved in the regulation of hormones. The hypothalamus is located below the thalamus at the base of the brain. It participates in homeostasis, including hunger, thirst, and sleep, and receives sensory information, including information from receptors inside the organs of the body. Thus, it is informed about changes in the organism's physiological status. The hypothalamus controls the pituitary gland, which is an endocrine gland attached by a stalk to the base of the hypothalamus. It is an endocrine gland because its primary function is the release of hormones. The pituitary gland is often referred to as the master gland because most of its hormones are tropic hormones whose primary function is to influence the release of hormones from other glands. For example, gonadotropin is a pituitary tropic hormone that travels through the circulatory system to the gonads, where it stimulates the release of gonadal hormones. Glands are organs that respond to imbalances in the body in order to regulate internal activities, such as the intake of food or water. They react by increasing or decreasing their production of hormones, which then travels to a target organ, where they lock onto specialized receptors on the surface of cells. This binding triggers a physiological change that restores homeostasis. The hypothalamus is the crucial link between the nervous system and the endocrine system, releasing hormones that, in turn, trigger the pituitary gland to either stop or start secreting its hormones. Here is a list of the hormones that are released by the pituitary gland, along with its function. For example, if we take the growth hormone, it acts on the whole body but is especially important for growth and development in children. Adrenocorticotropic hormone, or ACTH, triggers the adrenals to produce steroid hormones that control the stress response. Imbalances in the body are detected and corrected using feedback mechanisms or loops. Levels of a hormone within the bloodstream are gauged, and the information is sent to the control unit in charge of that hormone, which in most cases is the hypothalamic-pituitary unit. If the level of a hormone is high, the control unit responds by reducing the production of that hormone to achieve balance. If the level is low, the control unit initiates an increase in production. Feedback mechanisms are also used to trigger rare homeostatic functions, such as contractions during labour. And there is also a mechanism called negative feedback here. In response to a rise in blood glucose, the hypothalamus triggers a chain reaction of reduced hormone production that results in a fall in glucose levels, which restores balance. An example of negative feedback will be given later in this lecture. Let us now turn our attention to homeostasis. We'll look at it in some more detail, and specifically we will look at thirst, hunger, and the sleep-wake cycle. So what happens with thirst? Well, when the body's water levels fall, salt concentration increases and blood volume decreases. Pressure receptors in the cardiovascular system and salt concentration sensitive cells in the hypothalamus detect these changes. In response, the pituitary gland releases antidiuretic hormone, ADH, which acts on the kidneys to retain water and produce less urine. The kidneys secrete the enzyme renin into the blood, which, via a series of reactions, forms the hormone angiotensin II. This is detected by the subfornical organ, which is connected to the hypothalamus, which in turn activates more ADH-producing cells and creates the sensation of thirst, which leads to drinking. The decrease in blood volume and increase in salt concentration that are caused by dehydration have a detrimental effect on the body's biochemistry. The neuroendocrine system responds very swiftly with a series of chain reactions that stimulate the sensation of thirst in an effort to restore the balance in the body. And what about hunger? Well, the body maintains its weight at a set point by using hormones to trigger the sensations of either hunger or satiety. To stimulate the appetite, the stomach produces the hormone ghrelin, while fat tissues decrease their production of leptin and insulin. These changes signal to specific neurons, we will call them type B, to start producing more neuropeptide and a Gouty-related peptide, which stimulates eating. We refer to these as NPY and AGRP respectively. The production of these peptides also causes other neurons, we'll call them type A, to inhibit the production of the hormone melanocortin, which usually works to suppress the appetite. These signals are transmitted to the lateral hypothalamic nucleus via other neurons, which generates the sensation of hunger. To suppress the appetite, the body's fat tissues increase production of leptin and insulin. These hormones signal to neuron type B to inhibit the production of NPY and AGRP. At the same time, the increase in leptin and insulin trigger neuron type A to produce melanocortin. These signals reach the ventromedial nucleus in the hypothalamus, which creates the feeling of satiety. And what homeostatic mechanisms are involved in the sleep-wake cycle? Let's have a look. Within the sleep-wake cycle, the suprachiasmatic nucleus in the hypothalamus plays a key role. We refer to the suprachiasmatic nucleus as the SCN. Light levels are sensed by the retina, and this information is relayed to the SCN, which then sends a signal to the pineal gland. This triggers the release of melatonin, the hormone that tells the body when to sleep. At this point, the brain becomes less alert and tiredness starts to take over. When melatonin levels fall in response to increased light, the waking part of the cycle begins. The circle, shown on this sagittal slice of an MRI scan, pinpoints the pineal gland. It's a pea-sized gland located beneath the thalamus. It is responsible for the secretion of melatonin. So it's falling levels of light which trigger the production of melatonin, which forms a link between the external environment and the brain's sleep-wake cycle. We'll now have a look at the stress response. When confronting environmental demands, life events, people evaluate whether the demands pose a potential threat and whether sufficient adaptive capacities are available to cope with them. If a person finds the environmental demands taxing or threatening, and at the same time views their coping resources as inadequate, they perceive themselves as under stress. It might be the case that the individual does not perceive the environmental demands to be taxing, or that they perceive the environmental demands to be taxing but have adequate coping resources. If either is the case, no stress is perceived. So perceived stress is really a balance between the environmental demands which are placed on a person and the coping resources that they have available to deal with those demands. The appraisal of stress is presumed to result in negative emotional states. These emotional states may trigger behavioural responses, for example, drinking alcohol, smoking, and sleeping changes, or they might result in physiological responses, for example, increased blood pressure, secretion of corticosteroids, primarily cortisol, that put a person under risk for psychiatric illness, such as anxiety and depression. They also put people under a risk of physical illness, for example, immune system diseases. Stress also plays a very important role in the onset of cardiovascular disease. If we are wanting to assess someone's levels of stress, assessing each component of the stress process can provide a more holistic assessment of stress rather than assessing just one component, such as physiological responses of stress. We need to take every aspect into consideration. It might therefore be important to consider life events. So these are the things that are happening in the environment, so the environmental demands. So whether the individual has been exposed to certain environmental demands, as well as how these were appraised. So were they appraised in a positive or negative way, and to what extent this had an effect on the person's life. For example, very positive to very negative. The perceived stress is important because this is the amount of perceived stress the individual is under. And this is of course related also to the coping resources or mechanisms that they feel they have available to them. We also want to consider the physiological responses in the person, namely blood pressure and cortisol levels. We would want to consider their psychiatric status. So, for example, the extent to which the individual is feeling depressed. And we would also want to consider physical illness in the individual. So what, if any, physical illnesses is the individual suffering from? If we were only to take one example or one dimension of stress, for instance, blood pressure, we wouldn't really fully understand the amount of stress that the individual was under. The chief components of the stress axes, the hypothalamic-pituitary-adrenal or HPA axes, include the hypothalamus, specifically the paraventricular nucleus or PVN, the anterior portion of the pituitary, and the cortex of the adrenal glands. So these make up the HPA axes. In response to stresses, trauma, or a variety of pharmacological agents, cells in the paraventricular nucleus of the hypothalamus release corticotrophin-releasing hormone, CRH. CRH, travelling in a portal vascular system, binds to corticotrophins in the anterior pituitary gland, causing the synthesis or release of adrenal corticotrophin hormone, ACTH, into the general circulation. Circulating ACTH binds to receptors on the adrenocortical cells, resulting in the synthesis or release of cortisol into the bloodstream. The bloodstream then carries the cortisol to the rest of the body. Cortisol exerts its effects throughout the brain and periphery, primarily through binding to two known types of corticosteroid receptors, the glucocorticoid receptor and the mineralocorticoid receptor. Cortisol also provides negative feedback through receptors located in the hypothalamus and pituitary, thus keeping the HPA axis activity in check. Cortisol is quite an important hormone for psychologists because it plays an extremely important role in the stress response, which is highly involved in fears, phobias, and anxiety-related disorders. It is also linked with disorders such as depression and obesity, which are prominent in society and pose big challenges for psychologists. Cortisol is a glucocorticoid steroid hormone produced in the adrenal cortex. Cortisol is often referred to as the stress hormone. Though cortisol does help the body respond to stress, it also has other functions, including converting fat, protein, and carbohydrates into energy. Cortisol is released in response to stress and low levels of blood corticoids, sparing available glucose for the brain, generating new energy from stored reserves and diverting energy from low-priority activities, such as the immune system, in order to survive immediate threats. However, prolonged cortisol secretion, which may be due to chronic stress or excessive secretion as seen in Cushing's syndrome, results in significant physiological changes and is implicated in many common conditions such as heart disease, diabetes, and depression. And we mentioned Cushing's syndrome here. This describes the signs and symptoms associated with prolonged exposure to inappropriately high levels of the hormone cortisol. Signs and symptoms include rapid weight gain, particularly of the trunk and face, with sparing of the limbs. But cortisol levels are not stable throughout the day. They also change due to circadian influences, like many other psychological processes in the body. The cortisol circadian rhythm is distinct and is regulated by the main circadian oscillator pacemaker in the suprachiasmatic nucleus, which is located in the hypothalamus. Normal individuals without disease of the HPA axes have very low or undetectable cortisol levels at midnight that build up overnight to peak first thing in the morning. Cortisol levels then slowly decline throughout the day. And we might assume that cortisol levels would be at their peak in the morning, ready for the day for the flight or flight response. Although cortisol levels show a circadian pattern depending on the strength of the stimulus or stressor, cortisol levels in the afternoon and evening can be elevated above those of the circadian peak. This figure shows the physiological cortisol circadian rhythm. The figure shows the mean and 2 standard deviation values of circadian rhythm of serum cortisol concentration calculated from 20-minute samplings over a 24-hour period in 33 healthy subjects. Cortisol levels are measured in micrograms of lead per deciliter of blood. Deciliter is equal to 1/10 of a litre. Cortisol has a distinct circadian rhythm, with a peak occurring at approximately 8:30 in the morning and a trough, which is the lowest point, occurring at approximately 20 past midnight or midnight. The lines here represent the mean and 95% confidence intervals. And we can also see 2 peaks here and here, at around 6 p.m. and also at noon. And these represent meal-induced cortisol stimulation. So these peaks are representing meal-induced cortisol stimulation. Cortisol levels provide an indirect readout of central nervous system function or activity. An increase in cortisol levels logically implies that circulating adrenal corticotrophin hormone levels have increased as a result of the increase in activity of corticotrophin-releasing hormone containing neurons in the hypothalamus. The HPA axis is highly responsive, and the effects are robust enough that both circadian effects and stressor effects should be observable in the lab setting. Cortisol levels are elevated in patients with depression. The HPA axis and depression is something that's been well investigated, and it's an example of negative feedback mechanisms working to influence our mental state. Depressed patients suffer from hypercorticolism, which are elevated levels of cortisol. The reason for this is that it is thought that depressed patients have a decreased sensitivity to the negative feedback effect on cortisol specifically a decrease in responsiveness or number of cortisol receptors. Evidence for this comes from the dexamethasone suppression test, or DST. Dexamethasone is a synthetic, so man-made, corticosteroid that has a similar effect to that of cortisol, but is structurally unique enough that it does not confound the measurement of plasma or salivary cortisol levels when administered to patients. In the DST, a small dose of dexamethasone is taken at bedtime. Now, in a normal individual, the morning plasma concentration of cortisol, which would normally be at its highest peak, will be diminished due to the negative feedback effects exerted on the axes by the exogenously administered dexamethasone. Okay, so the feedback mechanism would have reduced the morning plasma concentration of cortisol in normal individuals. But in a subpopulation of depressed patients, morning cortisol levels remained elevated. And this finding suggests that these patients have a dysfunctional HPA axis negative feedback mechanism. So let's move on to anxiety disorders now and look at them in relation to the stress response. This is something we might like to consider in those with anxiety disorders. So temporary feelings of nervousness, apprehension, and even panic in stressful situations are normal and appropriate. However, when these anxiety reactions occur frequently in ordinary situations and disrupt normal activities, it is considered to be a disorder. The disorder might be due to a thyroid disorder, substance abuse, a stressful event such as bereavement, or a family history of the disorder. Whatever the underlying cause, it will involve disruption of neurotransmitters, and the effect will be disruption of the body's normal control of its stress response, so the fight or flight response. With anxiety disorders, either the stress response fails to turn off or the stress response becomes activated at inappropriate times. For example, generalized anxiety disorder is characterized by excessive, inappropriate worrying that lasts for at least 6 months. Panic disorder is characterized by sudden, unexpected attacks of intense anxiety or fear. And in response to stress, the hypothalamus stimulates the pituitary gland to produce adrenocorticotropic hormone, the ACTH, which we've spoken about. ACTH stimulates production of adrenaline and cortisol by the adrenal glands, and these hormones produce the fight or flight response. Activation of the body's fight or flight response produces widespread effects on the body. Normally, the response turns off when the stress disappears, but in anxiety disorders, the stress response may be oversensitive or may fail to turn off. And we can see here the physiological changes that occur as a result of the fight or flight response. And these may occur in anybody in normal stressful situations. But as we've said, in anxiety disorders, the normal fight or flight response may not be turning off properly. So they may continue to feel these physiological effects. Now, phobias are a disorder in which a stimulus, for example, a particular thing, certain activities or situations, causes or evokes persistent irrational fears which disrupts everyday life. Phobias include fear of spiders, arachnophobia, enclosed spaces, which was referred to as claustrophobia, fear of heights, which is referred to as acrophobia, and also fear of crowds and public places, which is referred to as agoraphobia. Now, the causes of phobias are not known for certain. Some suggest phobias run in families or are the result of learning. The main symptom of a phobia is an intense, uncontrollable anxiety when confronted by the feared object or situation. Although merely anticipating an encounter with the feared object or situation can also cause anxiety. So in several cases, there may be symptoms of a panic attack, such as sweating, palpitations, breathing difficulty, and trembling when the object or situation is actually encountered. And very similar fight or flight mechanisms are involved in phobias as they are as those that are involved in anxiety. So let's move our attention now to measuring the hormones. So how do we actually measure these hormone levels, these cortisol levels? Well, there are three main methods for measuring hormone levels, especially cortisol hormone levels. There's saliva and blood serum, and the third is urine. But there's also a new approach now to measuring hormones using the hair. Now, hair will give a very different measure of hormone levels than the other three methods. So rather than being a snapshot measure of hormone level, hair will give a measure of cortisol levels over a much longer period of time. Now, saliva testing is proving to be the most reliable medium for measuring hormone levels. The majority of hormones exist in one of two forms, as we said, free or bound. So 5% of hormone is free, and the other 95% is protein bound. It's only the free hormones that are biologically active or bioavailable and available for delivery to receptors in the body. So those which are protein bound do not fit the receptors and are considered non-bioavailable. So when blood is filtered through the salivary glands, the bound hormone components are too large to pass through the cell membranes. Only the unbound hormones pass through and into the saliva. So what is measured in the saliva is the bioavailable hormone, the clinically relevant portion which will be delivered to receptors in tissues of the body. Saliva hormone levels are expected to be much lower than serum levels, or blood serum levels, as only the unbound hormones are being measured. So it's just the hormones that are actually available to the body that are being measured. Now, saliva testing is an easy and non-invasive way of measuring hormone levels. And the collection procedure is simple. The individual spits into a tube. The tube is placed into a machine which then analyses the sample and gives a readout of hormone levels. Storage is also simple because the sample can be stored at room temperature in the refrigerator or freezer. And at room temperature, the sample will last between 7 days and 4 weeks. One huge advantage of this approach is that it's simple enough for the participant to do themselves with adequate instruction. Now, serum tests are unable to distinguish the protein-bound, so inactive form of the hormone, from its free and biologically active form. So it can only give a rough estimate of hormone levels. So why is this important? Well, there are clinical implications here. So for example, when healthcare providers measure serum hormone levels and prescribe hormone replacement therapy based on those results, patients are often overdosed. So if the patients are then tested using saliva, the results are extraordinarily high, and confusion results from a lack of correlation between the two methods. Now, the urine test gives us a picture of hormone levels across a period of time. Urine hormone levels reflect the average amount of hormone present across the 24-hour period. This is in contrast to the serum or saliva test, which gives us only a snapshot of the person's hormone level at that particular point in time. So we can see a table here showing the potential advantages and disadvantages of saliva, urine and serum testing. So saliva testing in most cases is considered to be the best and easiest approach for collecting samples. So saliva and serum give us the snapshot of cortisol, and then urine would give us just the average over a 24-hour period. Some of the advantages of saliva are that the sample can be collected at any time. Now with serum, yes, the sample can be collected at any time, but the individual cannot collect the sample themselves. And obviously with urine, the sample is very much dependent upon the need to urinate. Serum is also an invasive method. With urine, the circadian rhythm can't be assessed because it's obviously looking at an average over 24-hour period rather than a snapshot. And the serum can't distinguish between bound and unbound and the unbound hormone. So it doesn't show what's actually freely available for the body at that particular point in time. So there are implications there for the type of study that we might want to do. So the method that we use to collect the hormone sample might depend upon the research question that we're asking. So for example, if we wanted to look at the effect of a stressful situation, so one that we've designed, on cortisol levels, we might take saliva measurements to establish cortisol levels. This is useful if someone has a fear and we're introducing them to their fear stimuli following some sort of therapy. If it's the case that they're not likely to encounter their feared stimuli frequently throughout the day, for example, they're afraid of spiders, we might want to know their cortisol levels in response to the spider over the course of treatment, and therefore a snapshot of that moment is more important. Now imagine that someone is suffering from depression and we want to know how their cortisol levels, so their stress levels, are changing over the course of therapy or medication. Depression is something that the individual must cope with continually throughout the day. It's not just in anticipation of something or when a particular stimuli is encountered. So here we might want to know the overall cortisol levels throughout the day, which changes throughout the day due to circadian rhythm effects. So in this case, we might be interested more in the urine cortisol levels. Now, we mentioned Cushing's syndrome before. Now, clinically, the commonest use for salivary cortisol is measuring late-night salivary cortisol as a screening test for Cushing's syndrome. So that's when we would collect the sample if we wanted to screen for Cushing's syndrome, and also what method we would use. Now then, although collection of saliva is a relatively easy non-invasive process, there are circumstances where obtaining an adequate volume of sample may be difficult. So for example, with infants, the disabled, elderly people, and those who are critically ill, it might be difficult to get those samples. Other influences that might affect our sample are drinking and eating, brushing the teeth, and smoking. So all these things would affect our salivary cortisol levels. So if the person's ill or has recently had bad news, that will also affect salivary cortisol levels. And potential blood contamination with the oral cavity may result in falsely elevated levels. So this is related to this is obviously related to the salivary cortisol levels. So patients or participants should be checked for fresh cuts or abrasions in the mouth if you are collecting samples of salivary cortisol. So even vigorous toothbrushing that results in open damage to the gum might have an effect on the salivary cortisol levels. So there are some practical things here that you need to consider when you are collecting your sample. So in practice, what does all of this mean when collecting your hormone measurements? So when collecting salivary samples, what would we need to consider? So we'd want to consider the time of the day that the sample is being collected. So this would be important because of the effects of circadian cortisol rhythm. We'd want to know whether the individual has slept that night and for how long. What else they might have done prior to the test. So whether the individual has been exercising, whether the individual has had a drink or food prior to the test. So an acidic drink such as a fruit juice will reduce the pH value, giving an artificially high reading for cortisol in the assay, while water can dilute the sample. It's better that the subjects do not eat or drink an hour before sampling. So whether the sample is good enough. So this refers to is the collector that we're using to take this to collect the saliva from, is it saturated enough with the saliva? What is a sufficient amount of saliva? So that's something that might be specific to the type of instrument that you're using, and you'll need to consider that. And what other disorders the patient has. So do they have depression? Are they obese? These are things that you would want to take into account. Do they have Cushing's syndrome? And also, do you have ethical approval to collect the samples? So you will need ethical approval as with any study. And do you have an HTA license to store the samples? So what is an HTA license? Well, HTA stands for Human Tissue Authority. The Human Tissue Authority is a watchdog that licenses organisations that store and use human tissue for purposes such as research and patient treatment, post-mortem examinations, teaching and public exhibitions. So basically, most things where you're wanting to actually store human tissue. They also give approval for organ and bone marrow donations from living people. So a salivary cortisol sample might not seem like much, but if you're wanting to store the sample, then the organisation you're working for will need to get an HTA licence. So we mentioned a new method for measuring cortisol levels using the hair. So a recent study led by Dr. Laura Mananian and others found that hair can be used to create a retrospective timeline of exposure to cortisol. So finding a new non-invasive method to measure long-term cortisol exposure could have a major impact on our ability to determine the role of cortisol in the development of many common diseases such as vascular disease, diabetes and depression. Dettenborn in 2011 found an increase in hair cortisol levels over 6 months in medicated patients with depression. So what these studies show us is that cortisol levels can be measured over a much longer time frame than was previously possible. So previously, with our other methods, we could only look at a snapshot of cortisol levels or over a 24-hour period. So you might wonder, why bother using both the hair analysis to measure cortisol levels when we already have saliva tests? So the methods are actually giving us different measurements of the same hormone. So as we said, saliva cortisol levels are a snapshot of the free unbound cortisol levels that are available for the body to use at that particular point in time. Saliva cortisol level measured at 8:30 in the morning is likely to be very different to the salivary cortisol level taken at midnight the very same day. In contrast, our hair cortisol levels do not fluctuate daily. They fluctuate only at the rate of hair growth. So in an individual with long hair, alterations in cortisol exposure over time can be recorded. So the two approaches can therefore distinguish between, for example, acute stress in saliva cortisol levels, so acute being rapid onset and a short time course. We can distinguish between acute stress and chronic stress. So hair cortisol levels will be better for looking at chronic stress. Chronic is a persistent or otherwise long-lasting effects. So we can see value in these different types of methods for measuring cortisol levels. If you are wanting to understand more about how hormones work in general, I recommend that you have a look at this textbook by Brown. Other reading specific to cortisol measurements is up on Blackboard. I hope you've enjoyed this lecture. The end.